captioning, as always. Feel free to engage that option by going to the More button at the bottom of your screen and then enabling closed caption. We are recording. Any of you who might have been on the first session of the family series yesterday will know, to my great mortification, that I forgot to record the session. So I'm gonna, slap myself on the wrist. I'm glad we have the weekly webinar set up to automatically record, and I'm going to figure that out for the family series. So that is a shameless plug, actually. If you want the family series information, you should definitely come live because my own limitations caused a problem. That being said, we have slides available from yesterday's family series, and we'll continue to share information from that. But we'll put that aside for now and get back to today's weekly webinar. We're super excited to have Dr. Wilkins back with us this week to talk about biomarkers. And I'm so glad, Dr. Wilkins, that you have your slide up, because I wanted to make sure to mention the part of your title today that says you are Assistant Director of the KU-ADRC's Mitochondrial Genomics, Metabolism, and I wrote in big letters on my notes, biomarker core. So who better could fit to be a speaker for this very important topic today? I think I mentioned on our webinar last week and in our family series class yesterday, I was doing a social worker's job of explaining what biomarkers are and saying, if you want to know the accurate explanation of what biomarkers are, come and hear Dr. Wilkins talk on the webinar today. So I'll leave you, but a couple other things I wanted to mention. I don't think I've mentioned this when Dr. Wilkins has been our speaker before, and that is she is a member of the Alzheimer's Association's International Society to Advance Alzheimer's Research Treatment Group, and equally cool, the Alliance of Women Alzheimer's Researcher Group. So that's super exciting. We're very proud to have Dr. Wilkins on our team, and always exciting to see women advancing in STEM fields, and particularly with Alzheimer's, because we know women have greater incidence of Alzheimer's disease. So anyway, I'm just very glad to know that about you, Dr. Wilkins, and glad to share it with you on the webinar today. So with that being said, tell us, what are biomarkers in Alzheimer's disease?
I will do my best. Thank you, Kelly. I assume you can hear me? Okay, perfect.
Good.
All right. So I think we'll just start off by trying to answer this question of what a biomarker actually is. It's actually kind of funny when you think about it. The only real definition of a biomarker is a biological marker, which really is not helpful, right? What does that mean? So when I think about a biomarker, I think of it as a measure we can take that can understand like the status of health or disease. Some really simple ones are taking your body temperature and you have a fever. You probably have an infection of some sort of virus or bacterial infection. Taking a blood sugar. That lets us know what's going on with maybe someone who is diabetic. Taking a blood pressure, that is a biomarker of how your heart health is, and how you're doing. So these are all technically biomarkers. So they don't have to be a blood test. They don't have to be anything really complicated. It's just something that can tell us a piece of information that we need to know about someone's health or disease status. So more recently, the Food and Drug Administration, the FDA, has put out pretty strict guidelines, which is really good because there's a lot of research going on into developing biomarkers. These guidelines are really meant to have people who are doing research or using biomarkers think about what type of biomarker they're trying to design, or how a biomarker is being used. So these are the categories right now that the FDA recognizes. And so they can all mean different things, although there is a lot of overlap. So a biomarker may work for more than one particular area. So we have diagnostic, which, can hopefully answer the question of, do you have a disease or the disease we're looking at? Prognostic might answer the question of how long would you live with the disease? Predictive might try to answer the question of who will get the disease. Response would be, is this treatment working? Monitoring, is the disease stable, worsening, or improving? And then safety is looking at, like, is a drug treatment safe? So when you think about it, maybe there's some medications that people might take, and we need to monitor like their liver or kidney function while they're taking them. So those would be safety biomarkers. Risk, do you have a risk for getting the disease? And so risk and predictive might have a lot of overlap, for example. And so I think that, there are a lot of different categories that biomarkers might fall into. And it's important to think about that. So you've probably heard a lot, if you have been in the AD field for a while, or are doing any like research studies, or have a family member, or you yourself have Alzheimer's disease, you'll probably have heard of the term ATN. Those are generally what the biomarkers that are measured in Alzheimer's disease are. So what does that mean? So essentially ATN stands for amyloid, tau, and neurodegeneration. So we call this the ATN framework. And why do we care to measure these things? Well, as you might know, people who have Alzheimer's disease are generally have these amyloid plaques in the brain. So measuring amyloid allows us to try to infer about the plaque load, and also neurofibrillary tangles. So these are made up of a protein called tau. And so that allows us as well to infer about the status of these in the brain. And the neurodegeneration, of course, is to, help us try to understand how much, how many, the neuronal loss that's happening in the brain that happens in Alzheimer's disease. Because when we get these plaques and these tangles, your neurons, which are the main component of the brain that function to allow you to have memories, they die. And so neurodegeneration biomarkers tell us about that process. I'm here showing examples of imaging biomarkers, just because there aren't a lot of like fun pictures of blood and cerebral spinal fluid biomarkers. Neuroimaging is really pretty. We get these pretty images, right? And so these are some examples of how we might measure amyloid, tau, and neurodegeneration in imaging. So we have amyloid PET scans. So that would be where you would go in, gets injected with essentially a tracer that then binds in the brain to the amyloid that's present there. And then when we take the image using the PET scanner, we would get something like this. And here what this means is the more red, the more amyloid is present. Blues and greens are negative, yellows, reds are higher. So everyone has amyloid in their brain, so you're never going to see a scan with no amyloid. But what you can see is that in someone who has Alzheimer's disease, they get positive scans like this because there's a lot more of it in the brain on that PET scan. There are some tracers to measure tau, and I'll speak about this more in a minute, but none of them are currently FDA approved. The amyloid scans are FDA approved. And so what this looks at is actually the amount of tau in the brain. So you would go through the same process. It's just the tracer that is being injected via an IV into you is going to be able to bind tau instead of amyloid in the brain and show those levels. And so again, everyone has tau in their brain. It's just in Alzheimer's disease, there's a lot more of it. And so that's why you see more oranges and yellows. When we look at neurodegeneration, this can be done in two ways. So there's MRI, so magnetic resonance imaging, where we can just look at the general structure of the brain and we can start to see where there's atrophy happening. We can see that the ventricles are larger. There's atrophy occurring in different areas compared to, this healthy brain. We can also do some other PET scans that look at glucose uptake into the brain, and that tells us also a lot about neurodegeneration. So, how do we measure biomarkers in Alzheimer's disease? Well, I categorize things in kind of three areas right now. One is neuroimaging. That's kind of more of a direct route because we can actually take pictures of the brain and see what's happening. The other is we can take cerebral spinal fluid or CSF. That comes from a lumbar puncture, or the others are blood-based. And so there are pros and cons to everything that we have to think about, right? When I think about imaging biomarkers, they are usually less invasive. For an MRI, we don't necessarily have to inject you with anything. PET scan, we do. They're relatively accurate. However, when we think about this, they do have a high cost. They need a lot of expertise for reading those images and looking at them, and it's not necessarily available in all areas. So we think about this, especially in trying to make sure that we get care and these biomarkers available for people who are living in rural areas that may not have easy access to, a PET scan or an MRI. CSF or cerebral spinal fluid, again, is also relatively accurate, but a lot of people consider this highly invasive. I know it's not fun having a lumbar puncture done where we actually put a needle into your back and pull spinal fluid out from around your spinal cord. And it's also not always highly available because it does take a healthcare practitioner that is highly trained in doing this procedure. And so, those are the cons of it. With regards to blood, obviously it can be less invasive. It's not uncommon. People usually get blood draws all the time. And it does tend to have a lower cost associated with it. Phlebotomists are generally everywhere. But there is more variability in the outcomes. These tests are not as developed. And then there are also the question that we always think about, especially when we're doing research on biomarkers, is does it reflect what's happening in the brain? When we take your blood, is that actually telling us what's happening in the brain? And does it give us information that we actually are needing to understand the disease process? So I'm going to touch a bit on these three categories of what people are generally using in Alzheimer's disease right now. So we have like the neuroimaging biomarkers in Alzheimer's disease, again, follow that ATN framework, the amyloid, tau, neurodegeneration. PET scans are used a lot, like I said, to examine brain amyloid and tau. And they can also measure, as I said before, glucose uptake into the brain using a different radio label tracer called fluorodeoxyglucose, or FDG PET, in case you don't want to have to say fluorodeoxyglucose a bunch of times. And you can see some examples down here from an FDG PET or a glucose PET scan. And what we see here is really opposite of what we would see with amyloid and tau, in that it's actually reduced in someone with advanced Alzheimer's disease, which is why there's less red. There's also MRI, which can examine brain volume and a neurodegenerative state. So these are some biomarkers that sometimes are used clinically as well as in research. Cerebral spinal fluid biomarkers, again, are going to follow the ATN framework, amyloid, tau, and neurodegeneration. You can get cerebral spinal fluid amyloid beta levels. This is an established test with known cutoffs for what is normal versus not normal. And this is going to be very important to think about when we talk about blood-based biomarkers. These are FDA-approved tests. Both the CSF amyloid and the CSF total tau and phosphorylated tau levels are FDA-approved, and they can be ran at Quest Diagnostics and are probably also covered by most insurance companies. And so these are really, the ones that maybe a clinician would order in addition to neuroimaging. We also use these in research as well. But like I said, these are highly utilized in the clinic. Now, for blood biomarkers in Alzheimer's disease, these aren't as well established. So again, we follow the ATN framework of amyloid, tau, and neurodegeneration. So we can look at plasma amyloid levels, plasma total tau, and phosphorylated tau levels. And then to measure the N, or the neurodegeneration, we can measure two different markers. One is called NFL, not like the football, not the National Football League, but it's called neurofilament light. And then the other one is called GFAP, which is known as glial fibrillary acidic protein. So these are actually proteins that when cells in the brain die, they release it. And so it tells us, again, about that cell death in the brain. Again, these are not largely FDA approved and are mostly ran in research settings because cutoffs are not clearly established, but they're currently working on being established. There are some plasma-based phosphorylated tau biomarkers that I believe are close to FDA approval. So my talk could be incorrect in the next couple of weeks, just as an example. And NFL, plasma neurofilament light, is FDA approved as well. It's often used in other neurodegenerative diseases, including Lou Gehrig's disease or ALS. And so those two are pretty close. Amyloid and GFAP, those are not quite there yet. They're still, undergoing a lot of research efforts. But usually these blood-based biomarkers would not be used as heavily in the clinic as much as they would be in research, although we are, as a center, working to deploy these in clinic because they're cheaper, easier for people. I know I would rather have my blood drawn than have a lumbar puncture personally. Yeah, so we understand why we would really like to have these blood-based biomarkers. So I wanted to kind of finish off before I answer questions of what does this mean? And I really liked this little cartoon that I found that talks about these three different modes of biomarkers that we just talked about. So we have cerebral spinal fluid, plasma, which is also blood, or imaging, and what's available. So in the cerebral spinal fluid, what we see is amyloid beta 42 is actually reduced. And then sometimes we can take this ratio of different amyloid beta species as well, and it's also reduced. In the plasma, it's controversial, what we see in Alzheimer's disease, which is why these are not approved yet. And then in imaging, amyloid PET, there are two tracers available, and we always see higher levels. And then we have tau, so ATN here. And the CSF, we always see higher total tau and phosphorylated tau. In the plasma, usually we see total tau, but again, these cutoffs have not been established, but we're getting there. And then in imaging, we again have some tau PET tracers that are still undergoing research as well before that final approval. In neuroinflammation, as I talked about, we have neurofilament light chain. This is approved in CSF. It is approved in blood or plasma. And then when we want to look at this with imaging, that's when we would use MRI to look at that atrophy, or that glucose PET scan to look at what we call reduced or hypometabolism. So I'm going to end with this little plug before I answer questions of, you're probably wondering what research is being done at the KU ADRC on biomarkers. This talk right here that I'm giving is to set us up for August 8th, where you can tune in to find out about what research we're doing here at KU on biomarkers to try to push this field forward. So with that, I will leave some time for questions. It looks like we have some chat, so I'm going to open that up. Greetings. Hello, David. Thank you. So Lloyd asked, can you measure tau via blood sample? Question answered, yes. So yes and no. It's being used in research, like I said. I could be wrong in a couple weeks, but it does look like phosphorylated tau might be approved here shortly. And that might be something that like a Quest or LabCorp could do for you. Rosa's asking, wondering why a primary care provider said a CT has mild atrophy and not to worry because it's an aging process. Yeah, so unfortunately, we haven't been able to fix this for anybody who's aging. We're hoping someday that that's the case. But it is normal with aging to have some brain atrophy happen. Mild is normal. There's always some. I don't know. I can actually go back and see if I can show my picture that I had at the very beginning. See how it says normal atrophy versus bad atrophy. So there's always going to be some level, just like there's some level of amyloid in everyone and some level of tau. Always with aging, there's going to be a little bit of atrophy happening. So that is why they are saying that. So, all right. Any other questions? Don't make this that easy.
I'll put in another plug for the family series, Dr. Wilkins. In the slides that we had yesterday, I have some images from Dr. Townley, Ryan
Mhm.
Townley, and Ann Arthur that show a progression of mild atrophy. And I think it's marked 0, like this is kind of what we might expect for a normal brain. And it progresses from 0 to 4. And it's a really convincing picture of kind of
Mhm.
how things change. So anybody who signed up for the family series, you'll have that in your slides. And if you twist my arm a little bit, anybody who doesn't have that picture, I would be willing to send it out and share it with you.
Yeah.
So keep that in mind.
Yeah. Okay, so another question from Lloyd. Fun fact, my oldest child's name is Lloyd, so I love that name. Thank you. I am in a cohort study. 1 year, my spinal fluid said I had normal tau. The following year, my blood sample said that I had elevated tau. What should I make of that? Well, that's really difficult. So if you haven't had the same test done each year, it's really hard because I don't know that necessarily the CSF levels and the blood levels are equivalent. And that's why we're running different tests. And if it was elevated tau, it would be interesting to know which tau test, because there are some that are much better than others. So what I would really suggest that you do is follow up with whoever the provider was that did your disclosure on your biomarkers and maybe just ask them what they think about that. And it's definitely, if you're in cohort, I assume that you'll have another test probably done this year or next year. I think if I remember correctly, we're doing CSF every 2 years, or it might only be once at this point. I know we're trying to boost the amount of CSF that we're doing. And so I would just talk to whomever the provider is that did your disclosure and ask them, what to make of that. So, because it really depends on what test was ran.
Dr. Wilkins, I took note of what you said about how in a couple of weeks, everything you told us today could be different.
Mhm.
And I think that that is really such a gift of research that, we just keep advancing and keep learning more about how all of this works. And that maybe if I can go so far as to say that that will get us we might be wrong about it today, but eventually we'll get to more information that will lead to treatment, cure, prevention.
Yeah.
Good.
We like to see things moving forward.
Yeah. Yeah. Okay. Well, I hope everybody took note that Dr. Wilkins will be back with us in, what is that, about 3 weeks? Time goes by so fast. And so I'm very excited to hear about the research that she'll tell us about. I know slightly more than I think our audience does because I have the topic on my planning calendar.
Yeah. It's a plug for a study that we're enrolling for currently, where we're looking to take our biomarker research, so that we can develop, some novel biomarkers that might be more helpful with some of the clinical trials that are being ran currently. So, yeah.
Very good.
Excited to talk about it. Thank you.
Thank you so much, Dr. Wilkins. If anybody else has comments or questions, feel free to speak up. I will tell you before we wrap up that next Thursday is July 25th. And our speakers, we have 2 speakers next week, so 2 for the price of 1. And they will be Sarah May and Greta Foy. Sarah is from Down Syndrome Innovations, which is a longtime partner of us here at the KU Alzheimer's Disease Research Center. She's I should make sure I have her title correct. Executive Director, something important. She's part of the team over there at Down Syndrome Innovations. And along with her, we will have Greta Foy, who is a new employee at the KU Alzheimer's Disease Research Center. And Greta has Down syndrome. And Greta is working with us on outreach and engagement and research to help advance the work that we're doing here at KU toward treatment and cure and even an investigational vaccine for people with Down syndrome against Alzheimer's disease. So those 2 will be our speakers next Thursday. I'm excited to hear from both of them. Dr. Wilkins, there's a question in the chat you might want to, I hope you'll take a look at.
Yeah.
And
Brain
then
ischemia.
one more
Yeah.
thing, Dr. Wilkins, before I forget. So Sarah and Greta will be our speakers next Thursday. I won't be here, I'm very sad to say, but that is because I'm going to Phoenix, Arizona, to the National Down Syndrome Congress Conference. So I'm going to learn more about Down syndrome and Alzheimer's disease and all that. If I can jump on the webinar while I'm out of town, I will, because I'm curious about what they'll say. But otherwise I'll have to watch the recording. So stay tuned. We'll have more information in the next newsletter about that talk. Okay. Thank you, Dr. Wilkins. Go ahead.
Yeah, no problem. Yeah, so brain ischemia, it's not currently used as a biomarker. We are collecting in our cohort some data looking at brain blood flow for research purposes and seeing if we can incorporate that into a bigger biomarker network that would give us a lot more information about Alzheimer's disease. Where you right now would see brain ischemia used outside of Alzheimer's disease is someone who might be undergoing a stroke, things like that. It is used clinically for that reason. But with regards to Alzheimer's disease, it's not a biomarker of Alzheimer's disease, but we do see it in individuals who have Alzheimer's disease, and it is usually noted and marked in those neuroimaging, clinical notes. But we do have an entire what we call neurovascular research unit in the Alzheimer's Disease Center who are measuring things like that, brain blood flow and other markers of potential brain ischemia. So, yeah.
Great. David Beyer, are you waving at us, or are you wanting to make a comment?
Trying to formulate a question here.
Okay.
In doing research, I know as a researcher in astrophysics, we do cross studies, but I know that you said that the blood markers aren't as reliable. There's not as much data or something as the others, and perhaps it's cost prohibitive. But couldn't you, when you are doing an MRI or a CAT or a PET scan, and you see you collect data there at the same time, can you do a blood sample to see the correlation? And then the third one is I can't remember what the third one. Oh yeah, the third one is the spinal tap. Have those 3 correlated and follow those, or is that just cost prohibitive?
It's almost like maybe you should come work with us, I think. Yeah. Do you need a job? No, we actually do have studies that are doing that. And I was going to talk a lot about that on August 8th. So we have a study currently enrolling, not necessarily to look at ATN, but some of the other novel biomarkers. We're asking people to get a blood draw. We're asking them to get an MRI and a PET scan, and so that we can compare across. Yep. It's a great idea.
Longitudinal data. Yes.
Right
Okay.
now it's just one because our funding is limited to 1 year. But what we're hoping is that when that study when that study completes, and hopefully the data are supportive that the group who's investing in this effort, called the Alzheimer's Drug Discovery Foundation, will want to continue to invest so we can do longitudinal studies. Yep.
Very good. Thank you.
And there is one more question from Suzanne asking, is some amount normal for a 70-year-old? Yes. Unfortunately, there's always, with aging, there's always some amount of ischemia or atrophy that is normal for a particular age. So again, I'm hoping that someday we can fix all of that. Aging is another topic of research, beyond just Alzheimer's disease, but there are people actively researching aging in general. So.
Excellent. All right. Well, right at 2:30. Perfect. Thank you so much, Dr. Wilkins, and
No problem.
everybody come back to hear more on August 8th. 0808 will be a very special day, and not least of which is because we'll hear more about the research that Dr. Wilkins is doing. So come back then, come back next week, and we'll hear about Down syndrome and Alzheimer's disease and the link between them and the research that we're doing here at the ADRC to support everybody in terms of that. All right, I think that's it for now. I hope some of you will join us for the family series, which continues next Wednesday, the 24th. You can visit the link I posted in the chat to sign up to attend in person, which would be great. We have snacks, and or on Zoom. And I promise to record it so that if you can't make it, you still can watch it. All right, thanks a lot. Looks like everybody's pretty grateful to you, Dr. Wilkins, for your great explanation.
Oh, thank you. No problem.
This is recorded, so it will be available later today. I'm in a place where the internet is super fast, so it shouldn't take me long to upload it and get it posted online. All right. Thanks, everyone. Take care.
Thank you.
And a plug for the family series. I did it on Zoom yesterday. It was tremendous. I thought, my goodness, it was so thorough that first day in the series. I thought, what more could we discuss? But Kelly did a gorgeous job. So thank you for including us on Zoom.
Oh, you're so welcome, Amy. Thank you for that feedback. I really appreciate knowing that those of you who are out there in Zoom land could get the information. And I'm just glad this is recording so I can go back and watch that part again. All right, everyone. Take care. We'll see you next time.
Bye-bye.